6 major classes. The 7th classes which I have not written. Why? Because suppose this question comes in examination and you fail to write the 7th class, exempted, you will get full marks. But if you write the 7th class, good, better. Okay. So in each cases you have to give one general example and one specific example of each of the classes. So we will see. Then this nomenclature is another question very oftenly asked. Now, what is active site? Not much is asked as per the new syllabus is concerned, but a slight idea should be useful for your better understanding of the, say, future mechanism of action of many drugs. So when I say active site and of an enzyme, so it encompasses a very small region of the enzyme. The whole enzyme is like a classroom and the active site is a blackboard, like that, correct? So that active site contains certain amino acid residues which participate in the enzymatic catalysis, Now this active site again can be divided in two parts, like the substrate binding site and the catalytic site. Okay. So what is can anyone explain me what is the difference between these two, substrate binding site and catalytic site? As if I am writing with this pen, correct? Now to write, to the movement, these 3 fingers are sufficient, correct? For some, only 2 fingers are sufficient. Okay. What the rest of the fingers are doing here? They are supporting the other fingers in doing the same work. If these 2 fingers are not there, then the grip will become different and it will act differently, correct? Similarly, the substrate binding site is comparable to the palm, correct? And the catalytic site is comparable with the 3 fingers. Actually, the subunits or the amino acid residues which participate in holding or capturing the amino acid residues or hydroxyl residues or zinc atom or whatever it is, the substrate, correct? Fine. So the catalytic site is the portion where the catalytic action takes place. So it is smaller in size compared to substrate binding site, correct? Alright. So active site, then smaller substrate binding site, then much smaller catalytic site. Now how to determine a catalytic site in a given enzyme? Suppose the enzyme is a globular protein. Look out for residues which are located internally to the structure but hydrophilic in nature. Why? Because the basic requirement for a protein to be globular is that all the hydrophobic substances will be kept inside the structure away from the water outside, or hydrophobic fluid. So inside you must not find any hydrophilic residue. If you, then it must have some special activity. What is that? It is activity to hold the substrate in position so that the enzyme can act. Alright. Now how to determine the active site? There are many methods, few of them has been depicted here, but here the most important thing is the X-ray crystallography. You must have heard about this technique, right? This was a technique used for determination of structure of DNA, yeah? In 1952. 1952, long back. So it's a very old technique. Can anyone tell me what is the technique about? Imagine this is a source of X-ray and my hand is mimicking a molecule, correct? So this is the shape. Now you don't know how the hands look like. You have no idea how the hand looks like. You have never seen a hand, human hand. You have seen only the what? Shadow. Correct? See? Can you see different shapes? A molecule can exist in any plane. So the X-ray will give different shapes. So then it falls, the responsibility comes to the scientist or the discoverer to design the molecule first and then take the photograph, normal camera, and match from all direction and match with the shadow produced by the X-ray. Why X-ray? Because X-ray will take photograph of very small molecule and our normal camera will take the photograph of large molecule. So they started making the different ball and stick models in different shape and started taking photograph from different angle. Then they matched whichever structure showed the maximum match percentage with that of the X-ray crystallography shadows. That means the ball and stick model what they made had a structure similar to that molecule. Right? This is the way by which they used to do. And it used to take a very, very time consuming process. In 1952 there was no use of computer. There was no use of computer in the laboratory. So what they had to do? They had to make libraries of photographs. And painstakingly one person is to go and match every photograph. Okay? That's how they actually found out what could be the probable structure of DNA. So this is the process of X-ray crystallography. Correct? Why crystallography? Because first you have to make it crystal, otherwise the shape will change while taking the photograph. So reduce the temperature, make it solid, and then take the photograph. Fine. So now the same thing if you perform when the enzyme with the substrate. What did I say? You mentioned, try to recall the latest version of enzyme activity or mechanism of enzyme action tells that enzyme and substrate both change their structure to an extent so that they fit each other in best possible way. Correct? This is called transition state of the enzyme, transition state of the substrate, or the transition state where the enzyme and substrate best fit with each other. Okay? So in that phase, if you take a photograph, then you make out. Suppose you are going to a park, correct, with your friends, all female friends, and you are going alone. What is your name? So suppose Imdad has visited a park with many female friends, not girlfriends, female friends. Now, how do you know, how he will know that which boy is particularly attracted to a particular girl? How will you know? If they catch them hugging each other in the park and take the photograph, right? Similarly, which substrate and where the substrate will bind, how do you know? Leave the enzyme and substrate in a solution, they will bind, reduce the temperature, they will freeze, take the photograph. Correct? Wherever the substrate will bind, that region is the active site. And the active site will never allow any other substrate to bind. That means that boy and the girl are made for at least for few moments. Afterwards they might change. Yeah, because change is life. Yeah, life is change. We can change many things, but we cannot change our parents, and we cannot change the colleges and schools where we studied. So, this college you cannot change. These teachers you cannot change. You have to bear it. Fine. So, freezing the protein, taking the photograph will give the idea where in the enzyme the active site is located. Just keep this in mind. If it comes for examination, just write in your own words. You need not to give any experimental evidence. Right? Then use a specific inhibitor. How do you know which inhibitor will act? Correct? Suppose you have a friend, Imdad has a friend whose name is Imran, and he is visiting in someone's house, in a neighbor's house, and the neighbor has 5 daughters. Now the problem with Imdad is, who is the girlfriend of Imran? How do you know? Bring all the 5, take them to the park along with Imran, leave Imran free. What will happen? What will happen? You will leave other 4 except 1. Correct? So they designed several inhibitors and mixed with solution containing enzyme. Then added substrate. Whichever test tube gave least reaction, that means that test tube contains the enzyme and the inhibitor which completely inhibits the reaction. If the reaction continues, that means no inhibition. If reaction stops, that means there is presence of inhibitor which is most suitable. So now we know the molecule number 3, suppose, is the best inhibitor. Correct? Then we can bind, we can take a photograph hugging the active site. Then we can publish a paper telling this is an inhibitor for this particular protein. Then we can contact some people who are making drugs. Then they will make some drug, they will send it in the market, and then the representatives of the company will come to you, visit you, and tell you that this is this. Sir, if you add this drug, then this will happen, and this person will never feel hungry. Fine. You will start writing the prescribing the what you will get? A nice Europe trip next month. If Europe is old, take Africa. Fine. So this is how things happen. Now we come to another property of enzyme. What is that? Enzyme has 4 properties. What is that? Mainly made up of protein. Number 2, high reaction rate. Number 3, specificity, and number 4, scope for regulation. So specificity. Now absolute specificity, group specificity, bond or linkage, stereo specificity, and geometric specificity. These 5 specificity you have to remember. And each one, at least one example for each. Absolute specificity. Example. Glucokinase, or in our lab we use, glucose oxidase. So this enzyme is specific for glucose, only glucose, nothing else. Okay? It's called, that means it's specific for only one molecule and nothing else. Then group specificity. Hexokinase. What is the group it is specific to? Hexose sugars. So it will act on glucose as well as galactose as well as fructose. It is not aldose specific. Fructose is also hexose. So it is a wide specificity. Yes. And what is the fallacy? See, hexokinase is present in all cells. I am talking about human body only. And glucagon is present in only liver. Actually, glucagon is type 4 isoform of hexokinase. Hexokinase has what? 4 types. Type 4 is nothing but glucokinase. Right? So they have different function in different organs. Right? Bond or linkage specificity. Say, chymotrypsin, an enzyme secreted by pancreas, a proteolytic enzyme, is specific to the peptide bond where the C-terminal end, this peptide bond means like this, right? So it will act on a peptide bond where this group is donated by phenyl or aromatic amino acids. Okay? Understood? So it will not see whether the protein has come from fish, katla, tilapia, prawn, doesn't matter. Whether the protein or peptide, polypeptide has got what? Aromatic amino acid, which has donated his C-terminal carboxyl group to the production of peptide bond or not. Then it will leave that peptide bond. Alright? This bond specificity. All, everything. Yeah? Carboxypeptidase, bond specific. Aminopeptidase, bond specific. Then trypsin, bond specific. Okay? What else? Stereospecificity. L-amino acid oxidase and D-amino acid oxidase. There are 2 amino acid oxidase. This L-amino acid oxidase will act on only L-amino acid, whereas D-amino acid oxidase will act on only D-amino acid. Now L and D, they are stereoisoform. What about geometric isoform? What is geometric isoform? Correct? Cis and trans. Can you tell me any enzyme which function on trans but not on cis? See, the enzymes which participate in the formation of eicosanoids, they function on cis fatty acids. Cyclooxygenases or COX enzyme. They function on cis form of fatty acid, arachidonic acid. Trans form of arachidonic acid is not their substrate. Correct? The best possible stereospecificity could be given by, this is given right. Another example you can remember, that is citrate synthase. Another example. Okay. So that's all for this topic. Bolo. Trans example আছে beta oxidation, and there is a pathway called beta oxidation of fatty acid. The enzymes are present over there. They all function on trans form. They never function on cis. Whereas these enzymes, they always function on cis form. See, the cis trans form, one is oleic acid. This is cis form, and this is elydic acid. This is trans form. Same acid. Cis form has different name, different function. Trans form has different function. Like the same fatty acid, if it is present in trans form, what will happen? It will be harmful for the heart. Coronary artery disease. See, in the packets of the chips or fried foods, there is zero trans fat. Means what? So it contains zero trans fatty acid. Correct? So trans form of fatty acid are removed. Only cis form are given. But problem with trans and cis form is, if too many trans form or cis form is there, then what will happen? Suppose 10% cis form. What will happen? It will increase free radical generation in the body. So not only increase, we have to introduce cis form, but also we have to reduce the size of the fatty acid to make it less harmful. If it increase, it is good, but it is good in what? In preventing coronary artery disease. But it is bad in other sense. It will increase the generation of free radicals more. So what will do? We will take an optimum level. We don't know which one is best, so we will go for an optimum level. Okay. Then there are 5 ways of catalysis. Quickly write down. Acid base. আমি বলে দিয়েছিলাম আগের দিন, হ্যাঁ বলা হয় না? Acid base, electrostatic, covalent, metal ion, proximity and Orientation effect, and transition state stabilization. There are 6 ways by which enzymatic catalysis takes place. প্রথমে কি বলেছিলাম? How enzymes work. এবার কি বলছি? Mechanism of enzyme catalysis. These are two are different. Why? I give you an example. We all eat with hand. Chinese people use chopsticks, we use fingers. But both of us, both are using hands. Correct? So how enzymes act is like using hand. And what is the type of catalysis? Are they using fingers or something else? Or they holding something with the fingers? So catalysis is different. How molecular level, at the molecular level, how they are bringing up the changes, that is called mechanism of catalysis, and that is what we are going to study here. There are 6 ways by which we can do, correct? And there were 3 ways of explaining how enzymes work, but there are 6 ways to explain how enzymes catalyze, correct? Two are different, right? Because previously what I said, there are 3 things, only one is actually correct, rest 2 are not so correct. But here all 6 are scientifically proven. Fine? Now, acid-base catalysis is nothing but a mixture of acid catalysis and base catalysis, general acid catalysis and general base catalysis. So, wherever the enzyme will take up, act as an acid, means they will take up hydroxyl group or donate H plus ion, it is called acid catalysis. Wherever it will act as a base, that is what? It will donate hydroxyl ion or take up H plus ion, called general base catalysis. Wherever acid and base are there, so both are happening together, like oxidation reduction, there are always something is getting oxidized, the other thing is getting reduced. That is why you call it redox reaction. Both are happening simultaneously. Similarly, here also like donating a proton, general acid reaction, accepting a proton, general base reaction. If they actually function together, then we call it acid-base catalysis. Example, say you can say the histidine imidazole. What is there? The imidazole ring of the histidine has got extra hydrogen ion, so it can donate, so it can function as a base, sorry, acid, right? Like this. Here are some residues, amino residues, which can donate H plus ion and which can accept H plus ion. So, if you find in the active site glutamate or aspartate, that means this is a proton donor, and in this form it is a proton acceptor. Lysine and arginine, see the epsilon amino group will donate the H plus ion. So, that will be the what? General acid, and if they exist in this form with a lone pair, then it will accept one electron, H plus ion. So, then it will become proton acceptor, general base form. Cysteine, RSH, side group, the H can be given and it become RS minus. So, together 2 cysteine can join to form cysteine. Histidine, this is called imidazole ring, it can donate H plus ion here. Serine, the H plus of OH can be donated. Tyrosine, the OH of the this group, hydrogen of this OH group can be donated. So, these are 2 forms, okay. So, they can, these things are there, that means they are acting like acid base catalysis. If the active site or catalytic site of the enzyme contains these residues, they probably participate in general acid base catalysis. That is what I wanted you to remember. Electrostatic catalysis. Now you know there are 3 types of bonds, covalent and non-covalent, correct? Covalent can be of 2 types, normal covalent and coordinate covalent, correct? And the non-covalent is the electrostatic bond. Example, sodium chloride, where positively charged substances attract the oppositely charged substances, correct? And vice versa. So, if we find the enzyme and substrate are behaving in such a manner, so that one is positively charged another is negatively charged. And what else? In order to establish an electrostatic interaction between two moieties, oppositely charged two moieties, what is the most important things to achieve? The flow of electron or the static electricity or conductivity in a medium? How you get it? If the medium is vacuum, is it conductive? If the medium is full of water, is it conductive? Yes or no? Yes? No. Pure water, zero conductivity. So, first thing an enzyme must do, exclude all water from the active site. Then only substrate can come and bind and enzyme can interact. All reactions, mostly 99.99% reaction except hydroxylases, where water is added, everywhere water is excluded from the active site. Example hexokinase, example glucokinase, example PFK1, example PFK2, any except hydroxylase reaction. Correct? So, why? Because water has highest dielectric constant. So, you remove water, then current can be used for making a electrostatic bond interaction. Interaction means what? That interaction cannot be seen, it is invisible, but they will attract each other, just like gravitational force. Now, just opposite to this, covalent catalysis. When an enzyme and substrate interact, then there is a possibility that the substrate will try to go away. Why? They are not bound by any rules of society. There is no society among the chemicals, correct? So, they can go anywhere. So in sometimes enzyme has to hold it. Actually by bond, formation of bond. Those reactions are called covalent catalysis. How? Suppose they want to add one molecule of water. So what they have to do? Suppose they want to add one molecule of water to another molecule. First they have to hold the water, then only they can put the water, right? If you want to throw a ball, then what is the first thing you must do? First you get the ball, hold it, then only you can throw. So when you take the water, how do they take it? By forming a covalent bond. Example of such example, covalent catalysis, best example is carbonic anhydrase. Carbonic anhydrase, the enzyme which actually put water into plus carbon dioxide to form carbonic acid, where the concentration is higher, 10 times higher inside the RBC compared to plasma. Right? Metal ion catalysis, there are two ways metal ion can participate. One is metalloenzyme, another is metal activated enzyme. Remember the example of each one. Metalloenzyme example, carbonic anhydrase, metal activated enzyme, any kinase reaction. Glucokinase, hexokinase, how do they do it? This is an example of metalloenzyme. Covalent catalysis. Zinc, this is a part of what? Carbonic anhydrase. This is a zinc molecule which will hold the water first, then add the water to the carbon dioxide to form bicarbonate. Right? This is covalent catalysis. This is metal activated enzyme. See what magnesium is doing here? This is the first step of glycolysis, that is addition of phosphate to 6th position of the glucose molecule to form glucose 6-phosphate from an ATP in presence of magnesium ion and hexokinase. What does it do? These hexokinase and glucokinase, the enzymes, their face is like a shark. That means the active site is like this. The active site is located here. Correct? The active site is just has space just sufficient for one hexose sugar. Fine? So one hexose sugar is occupying. And it can accommodate only one phosphate. Correct? But the problem is these nose, overhanging nose of the shark head, it has got some residues. What residues? Which residues? Mostly hydrophilic. Correct? That means positive or negatively charged. Why? Because it is outside, away from the, towards the water, away from the core of the protein. Now, the phosphate is also negatively charged. So, what will happen? Can it enter in a charged space? No, it will be repelled. It cannot enter into the shark's mouth where the active site is located. So, what will happen? It has to reduce the charge over phosphate so that it become more neutral, and then it can enter to the depth of the active site. Clear? So, what will happen? The magnesium ion will shield these two phosphate, beta and gamma phosphate. That means the innermost two phosphates. Sorry, outermost two, beta and gamma. The innermost is alpha. Alpha, beta, gamma. See the structure? ATP. Right? So, the charge over beta and gamma will be reduced. We call it shielding, not reduce. We call it shielding the charge. So, presence of dication will shield the charge over beta and gamma phosphate so that they can enter to the center of the active site of the hexokinase or glucokinase where the phosphate will be transferred. Okay? Without magnesium ion, that cannot be done. That is the role of metal ion activated catalysis. Okay? And having zinc, which is fixed in the structure, is called metalloenzyme. These are few examples of metal ions and the corresponding enzymes. You can take a photograph, you can remember from any book, up to you. See, I will never tell you, you write this, you write that, unless it is very, very important. It is up to you whether you take it down, remember it or not. Question will be asked in the same way, answer will be the best possible answer has been given in the class, but you will forget after a week if you don't write and don't read it. Then you will take up the cyclostyle book and read something which has no meaning. You don't understand. You don't understand the head or foot, nothing, only the body. Up to you. Then factors affecting enzyme action. Alright. Just write down the factors affecting enzyme action. There are 6 factors. Many books have given more than 6. Again, it's up to you. My memory is short. I remember only this. Enzyme concentration. In each thing, you have to remember this one type of graph. So first you have to draw the graph, and then you have to explain how this graph has been drawn. This is enzyme concentration and this is rate of reaction. So rate of reaction is what? density. What is density? কি হচ্ছে একটা রিঅ্যাকশন দেখালাম সাবস্ট্রেট প্লাস এনজাইম এটাকে কি বলা হয় ফ্রি এনজাইম এটা হচ্ছে বাউন্ড এনজাইম বা এনজাইম সাবস্ট্রেট কমপ্লেক্স ইএস কমপ্লেক্স এটা আবার ফ্রি এনজাইম তাহলে আমি বলতে পারি রেট অফ রিঅ্যাকশন মিন্স হাউ মাচ সাবস্ট্রেট ইজ বিং ইউটিলাইজড পার ইউনিট টাইম or how much product is being formed per unit time. So কি বলবো how much substrate is being utilized per unit time minus dSdt or dPdt. How much product is being formed. Now which one to take? In any enzymatic reaction, this is the first question you have to answer. Because otherwise you cannot monitor the reaction. In some reaction, it is easier to monitor the substrate. In some reaction, it is easier to monitor the product. Okay? So for example, in reactions of glucose oxidase peroxidase, it is easier to monitor the product H2O2. Correct? In another reaction, estimation of AST, it is easier to monitor the substrate NADH. Correct? So it depends on the type of reaction what we are following, which one to take. Whatever is easier to monitor, we will take that. So whatever it is, in both the cases, V stands for rate of the reaction. Right? So more enzymes, higher the rate. Up to what? What is fixed here? What is fixed in this graph? The substrate. So the graph will continue go like this until the substrate gets exhausted. More enzymes, more reaction. 5 men can perform a work in 5 days. So 10 men will perform in work in 2.5 days. 15 men 1.75 days. Correct? 20 men less, further less, 1 day. Correct? But here work is amount of substrate. So if the substrate is constant, what will happen? After some time, however enzyme concentration you increase, it will not increase, it will become parallel. Why? Because all substrate has been taken up by the enzymes. Now there is no more substrate taken up by the enzyme. So even if you increase enzyme concentration, the rate will remain same, it will become parallel. So when you draw this graph, you must tell these lines what I am going to tell, and you need to take it down. যেটা বলতে যাচ্ছি সেটা তোমার লেখার জন্য হাত জোর করে অনুরোধ করছি, দয়া করে আমাকে না দেখে খাতায় দেখো। আর তুই হাই তোলা বন্ধ কর, এই ছেলে, ইউ, ইয়েস, টু মাচ। देखो, rate of reaction increases along with the increase in enzyme concentration. rate of reaction or velocity increases with the increase in enzyme concentration provided substrate is not a limiting factor. Provided substrate concentration by substrate is not a limiting factor, means a never ending supply of substrate. In that case, the graph will go on increasing like this. Otherwise, the graph will become parallel after some time. So, you need to explain by drawing this graph and telling where it becomes parallel and why it becomes parallel. That's all for today.